And when we look at the bilateral animals, there's two different types of bilateral animals. You're either a deuterostome or you're a protostome. Okay, there's bilateral, deuterostome, protostome. There's a big division there. And if we look at the words, and over here we are vertebrates, so we're moving over here. We're deuterostomes. And if you look at the word stome, whenever you see stome in a biology word, it technically means mouth. So it might be referring to the mouth. It might just be referring to a hole, though. Do you remember on the underside of a plant leaf, those little breathing holes that plants have? Those are called stomata. So it means hole. Technically, it means mouth. And deuterostome means number 2 mouth or second mouth. And protostome means first mouth. So that's what the words mean. Let's see where they got that from. Again, the details of this are not as important as the fact that this stuff exists, and we don't touch on a lot of really detailed physiology in this class, so this is one place I like to. So these are embryos again. And the difference between a protostome and a deuterostome is there's 2 different embryological pathways. And see, I told you I would never say embryology again, I'd start saying development. There's 2 different developmental pathways, and there are 4 things, 1, 2, 3, 4, that are very, very different between protostomes and deuterostomes, and they happen very early in the embryo. They're laid out in this chart here, but I'm going to go through them one by one. Cleavage means to split, to divide, not what you think it means. You're like a meat cleaver, right? And when biologists use the word cleavage, they're almost certainly meaning cells dividing. When a cell divides and they split, you did mitosis, right? We call that cleaving. This is the actual separation, cleaving. And the cleavage patterns are different in deuterostomes and protostomes at a very early stage. Let's start with deuterostomes. In deuterostomes, we get to a 8-cell stage, 4 cells on top of 4 cells, and you can see the cleavage furrow, that's called. It makes a nice straight line, a radial line. And then the embryo just keeps going. What protostomes do is when they get to that 8-cell stage, look what's the difference here. The top tier of cells makes a little half turn. And the cleavage furrow, the groove here, is spiral. That's called spiral cleavage. That's the old-fashioned way to do things. Deuterostomes don't do it. That's our ancestors' innovation, stopping this nonsense. That's a very easy thing to see when you're making embryos in a lab and watching the development. And it's a very distinctive feature. Protostomes are spiral, deuterostomes are radial. What does it mean? Don't know. It just, anything that happens that early is a big deal. It always makes me think of a Rubik's Cube. Look at this. See, do I have to click this? Yeah. These are probably sea urchin or starfish embryos. 2 cells? 4 cells? 8 cells? Look at the one over there. 16 cells without the turn. This is a deuterostome. I think that's cool. That's not, it doesn't happen that fast, that's time lapse. And there, we're on our way to forming a blastula, right? As this cells divide, it's going to hollow out. And that will be the blast. It's not a blastula yet, but it's going to hollow out. Hey, those are probably sea urchins. Another thing happens very early. Another thing happens very early on is something called cell fate. We've got the protostome, deuterostome version. Now, in cell fate, what that means is the cell is told what it's going to be as the embryo develops. And think back here. When the egg hit the sperm, the genome of this individual was set, right? Every cell in your body, outside your ovaries or your testes, let's just put them aside. Every cell in your body has the exact same genes as every other cell, right? You have the exact same chromosomes. Every one of your cells is a clone of the other cells. So, if this is you and that's the cell in your embryo, inside the nucleus there are all the genes necessary to make eyes, as well as livers and muscles. Even though this cell, its descendants may end up being part of the heart, in that heart cell are all the genes necessary to do everything in your body. But in your liver, the liver cells, all the genes involved with being a heart cell or an eye cell or a nerve cell, they're turned off and locked down and never used for your whole life. But they're there. The information's there. What cell fate is the cells get assigned a job. You're going to be part of the mesoderm, and any cell that's not, any gene that's not involved in being part of the mesoderm gets closed down and locked and never opened again. And in protostomes, the cell fate is determined very early in the embryo. At this 8-cell stage, these cells are already assigned their jobs. In deuterostomes, the assignment comes much, much later. And there's words for that. When the cell fate is determined very early, it's called determinate cell fate. When it's determined much later, it's called indeterminate cell fate. Okay? So in this embryo here, this protostome embryo with determinate cell fate, if you went in with a very sharp needle and broke that cell, destroyed that cell, you've killed the embryo. Because whatever that cell was going to be, the embryo can't make those things anymore. Whatever that cell was going to do, these cells can't do it. They have the genes, but they've been turned off. In a deuterostome embryo, you can reach in there and destroy that cell, and the embryo would say, "What the hell?" But it just keeps going. It doesn't matter. The other cells can pick up the slack from a much, much longer period of time. So, protostome, you take one piece out of that 8-cell embryo, and it's over. In the deuterostome, we can have twins. And what a twin is, an identical twin is one of those cells breaks off. And just, okay, I'm now 7 cells behind where I should be, but I can catch up. This embryo's fine without that cell. This one here just keeps going, and we get identical twins. Anyone here an identical twin? We've had a couple sets over the years. One set always sat in the back corner of the room, never made eye contact with anybody but each other. They wouldn't talk to each other. It was like I shouldn't say this, it was like a Twilight Zone episode. They were really, really sharp. Dressed the same. And we good on that? Remember, the body cavity is called the coelom. And again, these are details we're not going into because we're not talking about body cavities so much anymore, but the big deal here is protostomes form their body cavity one way, their coelom. Deuterostomes form in a completely different way. That's all I want you to know about. 2 different ways to get to the same thing, a hollow fluid-filled space. Finally, remember that opening in the original embryo called the blastopore? We need to see where that goes. Remember, you as a vertebrate and most other animals have 2 openings to your digestive tract, right? You got one and then one that forms later. One's the mouth, one's the anus, right? Okay. In protostomes, that means first mouth. That original opening, the blastopore, is the mouth, and the opening that forms later is the anus. And that's a protostome way. Protostome tells you that, first mouth. In deuterostomes, that first opening, the blastopore, becomes the anus, and the opening that forms later becomes the mouth. Second mouth. So basically, deuterostomes flip the embryo upside down. What does it mean? Don't know. But it's there and it's a feature. It's a very different way of doing things. So, like I say, protostome and deuterostome, there's two developmental pathways there. If we look at our evolutionary tree, again, we're working our way towards chordates, because that's the phylum of interest to us. Chordates are deuterostomes. We're done with protostomes now. We don't need to touch them ever again. Here's where we are. And boy, that's going to be a true false on the exam. True false, vertebrates or deuterostomes. And we'll see who gets it wrong. had a colleague who was on a doctoral committee. And when you get your PhD, you write your dissertation, and then you have these things called oral exams where your committee sits you down and grills you for about 3 hours. They can ask you whatever they want. They ask you about your research. They ask you about other stuff. And this poor kid was so rattled. He was having a really hard time. He was really bright, but he was having a terrible time answering the questions. I wasn't there. They were telling me this. And so one of his committee members said, just relax. Right? And answer me this. Are you a deuterostome or a protostome? And he was so rattled he cried. He didn't know.
Oh.
Anyway, we're deuterostomes. By the way, just as we finish here, there's the protostome groups. And there's the deuterostome groups. We're in a small club. We're represented here at Vertebrates Fish Day. We're in a fairly small club. So if we summarize, this is the vertebrate body plan. We're not worried about any of these other things anymore now. Yeah, of course we have tissues. Those evolved a long time ago. Bilateral symmetry evolved a long time ago. 3 germ layers a long time ago. Our body cavity is a type called a coelom, a true body cavity. And we're the cool way to go. We're deuterostomes. All right, did that. So again, focusing on us, chordates. There's these things here. This is just a review. You can get these and look at them later. Tissues, they evolved down here. Anything more primitive than that doesn't have them. Symmetry. Well, symmetry is a feature, again, the more advanced animals. Radial symmetry first. Jellyfish still show it. Everybody else is bilateral. Germ layers. Germ layers evolved here. Primitive way is 2. Cool things have 3. Body cavity, it just doesn't show. If you have a textbook, maybe 15 years old, they'll try to plot body cavities on this. It's very confusing. And then deuterostome, protostome. I think that's we have 2 groups that are named that, protostomia and deuterostomia. All right. I think I had this slide up here earlier, but it's worth reviewing again. Invertebrate and vertebrate. Vertebrate is a taxonomic category. It's a monophyletic group. There isn't, we come off, I erased it. There's an ancestral vertebrate, and everybody comes off, everything we classify as a vertebrate comes off an ancestor. And the vertebrate is just everybody else. Like I said last time, it's like you're an American or you're a foreigner. An American means a particular group. Foreigner is just the negative, not an American. So, and vertebrates are one part of the phylum Chordata. And everybody else, including some chordates, are vertebrate and invertebrates. Okay. This I found in the textbook. This was one of these slides. Move your finger around. Kind of interesting. It tracks the different types of animals and different body plan arrangements. Sponge up there, jellyfish here, and with different types of embryos. It's kind of an interesting approach. Put this on your screen and move your finger around a little bit and see if these things make sense to you. Some of those words I didn't use, but I think you can work your way through that. Okay. That's end of 8 and 9. All these PowerPoints are on Blackboard or Canvas, right? You're finding them, right?
Yes.
Okay. You certainly should feel free to print them off and bring them in and take notes on them. I rearranged a couple of them this morning before class, and if I rearrange them significantly, I do repost them. And if I repost them, it usually says updated. I delete the old one. It will have the same name, except the new version will say updated. And so if there's significant differences, you might check for that, but there's usually not. Let's get to living things in more specific context. And I call this chapter 23-ish. We're transitioning into primitive vertebrates, orange vertebrates, early vertebrates, and life evolved in water, and initially diversified in water. So I have a chapter called Living in Water, and later on as we transition from aquatic to terrestrial, I have a chapter called Living on Land. So some of the challenges about this particular environment. These are tuna, by the way. That's what a tuna fish looks like before it's in a can. So, like I said, vertebrates originate in water. And so this is pretty important. And I made this point earlier. The fundamental biology of the chordates or the vertebrates was laid down a long, long time ago. And even though you don't live in water anymore as a mammal, there's aspects of your construction and your physiology and your body plan that actually go back to these very ancient vertebrates. So when we understand the original plan, it's useful for understanding what happens next. So, water world. There's things to look at when we're thinking about water and living in water. And I make a list because I like to start with lists. Density, weight, and pressure. And one of the things we want to look at here is we don't live in water. So this is very, very different to us. And so it's worth giving this some thought. Density, for example, has a profound effect on evolution and body plan and how you interact with your environment. So a whale on a beach. Water is extremely dense compared to air. And so organisms that are evolved and adapt to live in water often have fairly different constructions than living on land. You have to deal with gravity. A whale, until he landed up on the beach, didn't have to deal with gravity in the same way. The density of water becomes a big issue. It's very supportive. It's also very heavy. And so, you're breathing, right? Your gas exchanges through lungs. Air doesn't weigh anything. That doesn't take any energy at all. But imagine if you have to pump water over gills. Water weighs a lot more than air does. And so that density becomes an issue. Well, in this case, weight becomes an issue. Ever gone fishing in a stream or waded across a stream? And if this guy steps 2 or 3 steps out here, just knee-deep in water, he's struggling because of the weight of that water pressing against him. But there are fish that are living in there and doing just fine. They have the adaptations to do that. And something we don't deal with as terrestrial animals is pressure. The pressure on the top of a 15,000-foot peak is a little bit different than it is down here, but not so much that you have significant adaptations for one or the other. But in water, look, we're at sea level here. And you remember your physics, right? Is this chemistry or physics? The amount of pressure pressing down on you is 1 atmosphere when we're at sea level. Every 10 meters you go down in the ocean or a lake, you're adding an atmosphere. So you just go down 100 meters, and that's 300 feet. And that's 10 times the amount of pressure you're experiencing here. So pressure builds up really fast, and creatures that live in those places have to be able to deal with that. Other things about water. Oxygen, solvent, salt balance is some of these things we'll talk about. Oxygen. All vertebrates have to extract oxygen from their environment. They either extract it from the air or they extract it from the water. Living in water presents some challenges that you don't face anymore because you're up on land. Like I said earlier, water weighs a lot more. So the actual mechanism for moving water over respiratory tissue is different. It's more robust. Other thing is water doesn't hold as much oxygen as air does. What's the percentage of oxygen in air, our atmosphere today? About 22-23% oxygen. There are aquatic systems where the oxygen level is almost 0, and there's things living there. So aquatic systems can be much more variable in the amount of oxygen they contain, and it's a little bit more challenging to get. If you're on land, no matter where you go on the planet, 22-23%. Go up on the top of mountains a little bit less, but not that much less. I was talking to, just complete aside, I was talking to Chris Wheeler, the geologist, and we were asking about the Carboniferous, which was about 300 million years ago. The planet was in the Carboniferous, and the planet was covered with huge rainforests, almost planet-wide. And that's when our coal beds were laid down. And with all that photosynthesis going on, a lot more oxygen going into the atmosphere, it's estimated that in the Carboniferous, the percent of oxygen in the atmosphere is 36%. That means if you got in a time machine and went to the Carboniferous and stepped out, you would faint. Now, when you hyperventilate, you get too much oxygen, you faint. That would be every breath you take. Like that. Instead, it's thought that at that level of oxygen, a tree branch falls off a tree and hits a rock. That's enough friction and heat to start a fire at 30% oxygen. Very different world. Now, the other thing, and we'll talk about this, is water is the solvent of all the chemical reactions in our body. You, as a terrestrial vertebrate, walk around as a self-contained aquatic system inside you. And you're separated from anything else. So you can maintain a very precise set of chemical conditions inside your body to do your reactions. But a lot of aquatic things don't have that luxury because they're exposed to the environment. They're exposed to an environment that's the same as their chemical reactions are occurring in. That becomes a challenge, particularly with things like salt balance. That thing we talk about, we're going to talk about this and we'll get to it today. I always think I'm going to get farther than I do. No, that's not going to work. Need a pen that works. The word for water and salt balance is osmotic balance. Or osmosis is the diffusion of water. So aquatic things have to worry about that. You don't. So the senses are, vertebrate senses were laid down in water, but the water world is different. You have these same fundamental set of vertebrate senses that have been readapted for working on land. So for example, light and vision, and when we'll touch upon this in a couple places, but aquatic things, many of them are highly visual. Well, there are aquatic systems without a lot of light. And light attenuates. It drops off as it moves through water very quickly. Go out in the ocean and you go down 100 meters or two and it's much darker than it is on the surface. So these are issues. Olfaction is the sense of smell. Gustation is the sense of taste. These are vertebrate senses that were laid down in these aquatic vertebrates, and they pretty much the same as they are in you. They just applied a little bit differently. Fish have the sense of smell, they have a sense of taste, but they are responding, well, the cues arrive at their sensors in water instead of transported on air, for example. The aquatic world is a noisy world, and hearing becomes an important sense. Sound travels really efficiently in water, much more efficiently than in the air, because water is so dense. And so hearing, very important thing. We've got lots of fishes that talk to each other, and you certainly are familiar with whales talking to each other, right? It's thought that the great whales, like the humpbacks and the blue and the fin and the sei whales, before we polluted the oceans with all that noise from boats and submarines and everything we do, it's thought that a humpback whale in California could probably talk to a humpback whale in Hawaii. That's how far the sound would have traveled. Now it's washed out with noise pollution. Then. Anyone take the whale class with Dr. Cartwright? No. Dr. Cartwright left us for Florida. Here's a sense we don't have. I call this etc. But it exists in aquatic organisms, at least some, and that's electrosensation. I put etc. there. There are some vertebrates out there that can taste electricity. I don't have the word for it. Feel, taste, smell. But electrical fields produced and radiated in the water, they can sense these things. All right, so any kind of aquatic habitat we have, I just put fishes up here. I need to broaden this into other kinds of vertebrates. But any kind of aquatic environment we have, we've got organisms adapted to deal with it based on those things we've been talking about. Now, there's some living vertebrates. we tend to think of aquatic vertebrates mostly as fishes, and that's my default thing because I'm a fish person, but we need to think broadly. There are other aquatic vertebrates out there. There's sea lions and polar bears and whales. And they're dealing with some of these challenges. They're dealing with all these challenges. Some do it in different ways than others. We'll look at the differences as we move along. Let's talk about salt balance and osmotic balance. And this, I reworked this the other day, so let me look at where I'm going here. Do you remember that lab in 201 where you had those little dialysis tubes and you put them in sugar water or you put them in distilled water? Remember you put the drops of sheep blood into distilled water, salty water? That's what we're talking about. That lab was to try to get at this kind of problem. When you're living in an aquatic environment, if you're exposing to that environment sensitive tissues across which diffusion can occur, things are going to diffuse into your body or out of your body, according to laws of physics. Let's look at that. We have three, I'm breaking them down into three aquatic conditions, three environmental conditions that you can deal with. And they're pretty basic. Saltwater, seawater, marine. Here you're living in an environment where the surrounding environment has high concentrations of salts. What do we call things dissolved in water? What's the word for that? Think back to chemistry. Isn't it solute? So the solute level is high. The salinity is high. What do we mean by high? It's higher than you are. Your internal tissues are less than the environment. Freshwater systems are systems that are not salty. So there are not a lot of solutes in the water. Here, you're living in here, your internal conditions are saltier than the environment around you. The solute level in you is higher than the environment around you. We've got freshwater, we've got marine water. And the third one is a very flexible one. That's in ecological terms, we call that estuary or estuarine. And that is where freshwater meets saltwater, like a bay or a harbor or a river mouth. So we've got creatures living in each of these types of habitats. And the problem is diffusion. Remember, diffusion is the movement of something from an area of greater concentration to lesser concentration, right? If we stick a membrane in there, some things can diffuse, some things that can move through the membrane will diffuse through the membrane. But if you've got things that can't get through the membrane, you can maintain a higher concentration on one side. Right? Okay. Osmosis is the diffusion of water. I looked and looked and looked. I found this little cartoon. Osmosis is the diffusion of water. And what we have here is a membrane. You recognize a membrane? A cell membrane. This is in a fish gill. Here's the outside. Here's the inside. The water molecules are little things. The blue things are the salts, or the green things are the salts. Here's the deal with osmosis. Water can diffuse through membranes, no problem. Water will always be able to get through a membrane. But there are a lot of things that are dissolved in water that can't get through the membrane. So they can't diffuse. So you can have more of something on one side, the higher concentration of solutes here than here. If they can't get through the membrane, they won't diffuse through. But water can always diffuse through. So when we have this imbalance, high solute, low solute, the solutes can't get through the membrane. And nature hates an imbalance. So it balances itself out the only way it can, and that's movement of water. And you got to think a little bit back. My dad had this word back-asswards. You got to think of it in reverse. If the solute concentration is high here, that means the water concentration is lower, right? And if the solute concentration is low here, that means the water concentration is higher. Does that make sense? The solutes can't move. Water can. And so when we have an imbalance, the water's going to go in the direction of the higher salinity. That's the problem aquatic organisms face. Whether this is the outside or the inside. If your internal conditions are different than the external conditions, water is going to move in the direction of the higher salinity to try to even out the concentration. In this case here, higher salinity, you can see the water is going to move through that membrane. Do you remember that experiment with the sheep blood? You put the drop of sheep blood in salt water, and they shriveled up because the water diffused out of the blood cells into the environment. You put those sheep blood cells in distilled water, and the water diffused in so fast the cells exploded. Remember that? So that is the issue here. We have a membrane that is selectively permeable. Water can always get through, but bigger things can't. And so I think, again, to summarize it as easily as I can, water is going to move towards higher salinity. You can't stop it. So there's those blood cells again. You throw the blood cell in the distilled water, and the water diffuses in so fast that there's no salinity up here, high salinity in here. Water's going to diffuse in so fast the cells explode. If you put those cells in salty water, saltier than the inside, water's going to diffuse out, and the cells shrivel up. And if it's equal, the diffusion in equals the diffusion out. Am I reviewing stuff we already know? Mm-hmm. Okay. So, this is the challenge that aquatic organisms face if they're exposed to the environment. And invertebrates are, and fishes are. But we have to consider aquatic vertebrates here. There's a mammal, there's a fish. Here's all the fishes. They're swimming in water, they're breathing water. And then here are the terrestrial vertebrates over here. We call them tetrapods again. Some of them have gone aquatic. Aquatic tetrapods, like all these here, these are all mammals and reptiles. Rearrange. Let me see where I'm going here because I've rearranged these slides. They've evolved to live in aquatic systems. Fish have never left aquatic systems. These guys have re-evolved to deal with that habitat. All yours. Whales come from terrestrial animals. So, tetrapods that have gone aquatic, like whales, are actually terrestrial animals that have reacquired aquatic adaptations. They're basically swimming terrestrial animals. And living on land gave them a certain suite of adaptations, including one really important set of adaptations. And that is, tetrapods are watertight. That's a water conservation mechanism. And part of the evolution of successful terrestrial living was a watertight skin. So when a tetrapod goes back in the water, he's not exposing anything to the environment. He doesn't have to deal with diffusion in, diffusion out, because he's watertight. Where is it? Fish, on the other hand, they're not watertight. Things are diffusing in and out through their skin. They have to move water over their gills to breathe. There's a tremendous amount of diffusion in and out through their gills. So fish have to deal with this stuff. He doesn't. You don't. When you jump in a swimming pool and swim for a couple hours, you don't face an osmotic crisis at the end of the day. Because you're watertight. Isn't it, goofy polar bears? So tetrapods don't have the same osmotic issues that fishes have, or invertebrates by any means. Although some tetrapods do. There are places on our planet where tetrapods live, and the only water available to them is seawater. So they may not have water or salts diffusing in and out of their body through gills, but they're drinking seawater. Whales are drinking seawater. So they have osmotic issues that way. but not quite the same. Can you drink seawater? Not if you hope to live. You don't have the mechanisms to deal with all that salt that you're taking in. Let's look at how fish deal with it. Let's see, where am I going with this? Did that. Fish are fully aquatic. So they are at one with their environment. And what I mean by that is they're not watertight. When they're swimming around in there, they've got sensitive tissues exposed to the environment, and if there's going to be diffusion, unless the inside is exactly equal to the outside, there's going to be water movement. So how do saltwater fishes deal with it? If you take a cell that has no adaptations for this particular issue, and a blood cell is adapted to one salinity, your blood, and you throw it in seawater, it shrivels, right? From another cartoon. So, without adaptations to deal with this, tissues and cells in seawater are going to be losing water to the environment. And that's the problem saltwater fishes face. Freshwater fishes have the exact opposite issue. Freshwater fishes, their cells have lower salinity levels than the environment, higher salinity levels than the environment, and so water is pouring into their bodies, into their cells. And unless they have adaptations to deal with that, they would explode. That's why you can't take a freshwater fish and throw them in your saltwater aquarium, or vice versa. They can't handle the sudden change. So there you go. How do we deal with this? We have three types of fishes, which we haven't done this in a lot of detail. We have jawless fishes, cartilaginous fishes, think shark. We have bony fishes, the bones that get stuck in your tongue, in your teeth while you're eating fish. There's different mechanisms here in these different groups of organisms. This is, let's see, I'll rearrange this, let me see where we're going. Did that. Okay. This is a little dichotomy that I noticed once, and we'll call it, we'll name it after me one day when it becomes a principle in textbooks. When you look at the anatomy of these different kinds of fishes, it corresponds to the position on the evolutionary tree. Jawless fishes have a certain anatomy, cartilaginous fishes have a certain anatomy. So the anatomy corresponds to evolutionary groups fairly nicely. But physiology, I don't know if physiology is more flexible or involves more recent evolutionary events, but when we look at the physiology, your way of dealing with the ecology of your particular environment, this is often more with habitat niche. And I think it shows up nicely in how different types of fishes deal with this osmotic balance. Their anatomy defines them as a primitive fish or an advanced fish. But we don't necessarily have a primitive way to do osmotic balance. We just have a saltwater way to do osmotic balance, or a freshwater way to do osmotic balance. So the physiology, I don't know, maybe we'll look at it more flexible, evolutionarily speaking, or more recent evolutionary events. They're adjusting to their environment. You can adjust your physiology more easily than you can adjust your anatomy, I guess is what I'm saying. So we got these niches, marine, freshwater, or both. So like a salmon that swims back and forth. The physiology of different kinds of fishes, like I said, we don't have a primitive jawless fish way to do things and an advanced bony fish way to do things. We have a freshwater way to do things and a seawater way to do things and maybe a moving back and forth way to do things. So physiology correlates with your niche in many cases. And so you have the challenges of the niche that you're in the seawater. That's the challenges you have to face. And then the organisms, as they evolve, respond with what they have. They respond with the molecular biology or the structures of the anatomy they have. And what they have gets modified to deal with this. So if you're a cartilaginous fish, natural selection modifies your cartilaginous fish biology to deal with living in seawater. If you're a bony fish, well, you've got a different set of tools to be modified for that. Let's look at how they do it. It's called osmotic balance. Yeah, we'll do this and then we'll take our next break. Did that right. Okay. Fishes, we've got all these different groups of fishes, primitive and advanced, but we have 3 basic strategies to deal with that. Hagfishes, we haven't talked about this yet, I need to work this in sooner. Hagfishes are the most primitive vertebrate we still have with us. They're at the very bottom end of the vertebrate world. They're jawless fishes. And they have the hagfish way of osmotic balance. There's the shark way. These are called cartilaginous fishes. There's a shark way of dealing with osmotic balance, and a few others do that too. And then there's the bony fish way of dealing with these osmotic challenges. So 3 different groups of fishes, 3 different sets of strategies. One of the easiest ways to deal with this challenge is to try to minimize your exposure. A whale has no exposure because it's watertight. But fishes don't have that option, but they can minimize their osmotic exposure. Fish scales are one thing that's thought to be important in that. That makes it harder for things to diffuse in and out. And really primitive fishes had heavy bony armor, and one thought is that's why they have it. It was an osmotic adaptation. But it's impossible to completely eliminate your osmotic exposure for your fish because you've got to move water over gills. And gills have to have very thin walls, very thin cell membranes, because you're going to get oxygen and carbon dioxide are moving in and out. So there's tremendous osmotic exposure here. If things can diffuse in, they will. And if things can diffuse out, they will. And what's diffusing in and out mostly? What's the thing that moves? Water. So a marine fish is hemorrhaging water into the environment through its gills, and a freshwater fish is just getting water moving into its body through its gills. The most primitive living fishes are called hagfishes, and they say, screw it, we're not even going to fight it. And hagfishes, the word for them is osmoconformer. If their environment gets more salty, their body tissues just get saltier. And if the water becomes diluted, gets more fresh, their body tissues just become more fresh. They have no adaptations to fight it. So whatever the environment is, that's what they are. That limits them to a very narrow range of salinities in seawater. But it means they don't have to fight it. That's the most primitive way to do it, the old-fashioned way, probably.
But can they live in the fresh water?
Nope. They have very little flexibility. That's the downside. The upside is they don't worry about it. The downside is they're stuck in seawater. Good point. Now, what a freshwater fish does, remember the problem with a freshwater fish is water's constantly pouring in through their gills and through their skin. So the freshwater adaptation says if they had no adaptations, they would explode. The freshwater adaptation is they never drink. Ever. And their kidneys are well developed and are pumping water out constantly. Freshwater fish are constantly peeing incredibly dilute urine. Water's coming through the gills. When they eat food, they swallow water, but they never drink water on purpose. They eliminate that water through their kidneys. A marine bony fish is dealing with the exact opposite problem. They're hemorrhaging water from their gills every time they take a breath. From their skin, water's leaving. So a marine bony fish never stops drinking. they constantly have to replace the water that they're losing by swallowing water. But what are they swallowing along with the water?
Salt.
Lots of salts. They have special structures in their gills that pump salts back into the water. That takes energy. It's expensive. It's their only choice. So they take in water. They're constantly drinking and constantly losing water. They're taking in far more salts than they need. They're pumping them back into the ocean. And they produce very, very little urine. Marine fish hardly ever, they don't pee at all, or very, very little. They don't waste water with urine. So, same kind of fish, bony fish, two different ways to deal with the problem, or two different problems and they're dealing with it. Couldn't find a cutesy graph for a shark, but sharks have a third way. Most sharks live in the ocean, so their problem is they're losing water to the environment. They drink, they take in more salts than they need. The sharks have gone a different route. What they do is, and all these words here we don't need, but what they do is they fill their blood with salts. They raise the salinity of their internal environment. So their internal environment becomes much closer to seawater. So the difference isn't as great. So they're not hemorrhaging large amounts of water in the environment, they're gaining lots of water because their internal environment is very close to the ocean. In fact, they make their internal environment so salty, their internal environment saltier than the ocean. They're more like a freshwater fish than a marine fish. And extra salts they take in, they have a special gland to pump salts back out of the ocean. So, freshwater fish, bony fish, never drink, pees all the time. Marine bony fish, never drinks, never pees, or rarely pees very little, and pumps salt back into the environment. Shark just plays with diffusion and elevates the salinity of their tissues to near that of the ocean, and it reduces the problem tremendously. And again, it's worth pointing out, here's aquatic tetrapods. We're waterproof. These are problems we don't deal with. We don't have gills when we swim. We don't have skin through which water can diffuse or salts can leave us. So we don't have specific adaptations to deal with this. Why? Because our skin is watertight and we don't have gills. So, unless you're drinking seawater, your exposure to seawater is fairly minimal when you're swimming around in it. It doesn't have quite the same osmotic problems as a fish does. Although some tetrapods have no choice but to drink seawater. It's the only water they have, and they have to have adaptations to deal with that. They drink the water, it gets far more salt than they need. You ever notice seagulls? Ever been close enough to a seagull? And they always look sick. You notice because their noses are running? No? Look at them next time you're close up. They have glands in their nasal cavity that takes salt out of the blood and it runs out their nose in sticky yellow mucus. That's why they look so disgusting. So it looks like their noses are running, they're crying all the time, but that's salt elimination. So they do have to deal with it if you're drinking seawater. I don't know how whales do it. The amazing thing, someone talking about whales once, and Dr. Cartwright told me, is whales, the right brain, left brain stuff. Whales can sleep with half their brain can go asleep while the other half is running the body. And then they switch. I think that's cool. Anyway, we go down osmotic balance. Okay. Let's take another few minutes here. Put your pencils down for a few minutes. And let's talk about it, and we'll move on. And we'll call it a week. I like it much harder.
So, where are you taking? Where are you taking?
We went to the Paradise Club, like the cafe there. It was really nice. It was beautiful. It was really busy. So it was fun. I'm sure we could